Much of the information that I'm getting ready to talk about is on the final, so definitely make sure you're paying attention to this. But more importantly, I want you to learn something from what's going on in synaptic transmission, what's going on, when one neuron is releasing neurotransmitters and causing a change in another neuron, what's actually happening. Okay, going into this, we should understand what the central nervous system is and what the peripheral nervous system is. And of course, central nervous system is brain and spinal cord. Peripheral nervous system are to the periphery of it, meaning the cranial nerves coming off the brain and the spinal nerves, the 31 spinal nerves coming off of the spinal cord. Okay, then the difference between the somatic motor system and the peripheral autonomic nervous system. We know the somatic motor system is the information going to our skeletal muscle cells that we have conscious control over, whereas the peripheral autonomic nervous system is basically the nervous system going to the internal organs, usually to the smooth muscle, whether that organ be something in the digestive system or whether that organ be a vein or artery, as particularly arteries, and it control the smooth muscle of the arteries. Then finally, we need to understand what glial cells are. And of course, they are the support cells. They are the nervous tissue support cells that support what the neurons are doing. A Schwann cell is something we talked about in the past as a good example of that. Of course, the Schwann cell is what became the myelin. So here we have a synapse, and specifically the space is the synaptic cleft. The cell releasing the neurotransmitters is the presynaptic neuron, and the cell receiving the neurotransmitter at its dendrites or its nerve cell body is the postsynaptic neuron. Now I'm going to click on this, or I'm going to show you the action potential traveling down, because remember, action potentials are only generated in the axons, but their purpose is to travel down and change the voltage inside of this bulb so we can allow calcium to come in.
Neuron to produce an action potential. An action potential in the axon terminal causes voltage-gated calcium channels to open and calcium to enter the terminal.
Now, again, this is exactly the same thing we described at a neuromuscular junction. If we take a little segment out and look at it, we see our phospholipid bilayer membrane. Here we see our voltage-gated calcium channels, and they're closed until this action potential changes the voltage by moving down there. Once the voltage gets here, the voltage causes them to open up and causes calcium then to enter into the cell, which triggers our little protein, our motor protein, to grab the vesicles, drag them down, fuse them with this area here, and release the neurotransmitter, or go through what we call exocytosis. So we saw our action potential come down. We saw our calcium voltage gates open up, calcium went inside, triggered our motor protein to pull our vesicles full of the neurotransmitters and fuse the membrane-bound vesicle to the end membrane, releasing the neurotransmitters into the synaptic cleft. Here's our postsynaptic membrane. Here's our chemically gated channels, the ones I like to call the key doorways. Here's a neurotransmitter that's been released, and it's going to go to the key doorway to open it up and then to allow ions to flow in. Now, this would be the direct method. If we had an indirect method, remember, the key would still fit a doorway, but it would cause a cascade chain of reactions to eventually open or close an ion channel, after a sequence of events where this is the first messenger, and it would trigger the release of the second messengers. Okay, so here we see the direct, again, we see the key, the neurotransmitter, open up the door. Now we see our ion flow, which is going to cause a depolarization or a repolarization inside of the postsynaptic cell. Now in this case, if it's sodium moving in, since that's a positive ion moving in, then it's going to cause a depolarization because it's going to cause the voltage to rise. And ultimately, we're trying to get the voltage to rise to threshold, at least the threshold at the axon hillock, so we can generate an action potential from the postsynaptic neuron. Now again, notice that according to the type of channel, type of neurotransmitter, and does that channel, is that a channel that's going to allow sodium in or another positive ion such as calcium in? We'll talk about that happening in the heart next semester. So if a positive ion is coming in, again, we're going to depolarize. But if we open up a doorway that allowed a bunch of a positive ion out, then that may cause it to hyperpolarize or drop below our resting membrane potential. The other thing that may cause us to hyperpolarize is, let's say, if this doorway was a doorway for the neurotransmitter GABA to open it up and chloride ions came in. Well, since chloride is a negative ion, then it causes my voltage to drop. So again, if I drop my voltage below resting membrane potential voltage, which in neurons is around -65 to -70, if I go below that, I hyperpolarize. If I go above -70 towards threshold, which is normally -55 to -40, then I am depolarizing. So again, opening up the neurotransmitters will either help us depolarize or hyperpolarize. It's according to which channel it is and what the neurotransmitter is. Now, according to the type of neurotransmitter, there's a couple of things that can happen to it after it does its job of opening up the chemically gated channel. Okay, it can either be broken down by enzymes. We actually saw that with the neuromuscular junction. Acetylcholine was broken down by acetylcholinesterase, and then the breakdown products were taken up by the presynaptic cell and repackaged, resynthesized, and then repackaged. Or it can actually be pumped into another area and potentially repackaged. So or it can be pumped directly back into the presynaptic cell. So, we can, and here what we're seeing is this situation where we're actually pumping, once we use that neurotransmitter, we're pumping it back in here and then forming a membrane around it and reusing it for the next time the action potential comes down, allows calcium to come in and trigger our motor protein to pull that. Okay, but the bottom line is we need to understand there's not just one way that we get the neurotransmitter off of the chemically gated channel to get it back into the cell. And sometimes it can even be transported to another cell, a glial cell, and then the glial cell transport it back. Okay, but here in this picture we do see pumps where we pump it from the chemically gated channels back in and then reform it. Okay, you will need to understand each of these steps and be able to identify a picture like this that actually shows which step we're at. I'll give you 2 steps per picture. I will put the pictures in order that they're supposed to occur, but then you will match the step with what's going on in the picture. Now, in this picture, we're already actually down here to the last step, but let's talk about what were the steps that led to the last step. Well, the first thing that had to happen, a neurotransmitter had to come down from the axon hillock all the way down the axon and then eventually here to the axon bulb. When that got here, then what it did, it triggered the opening of calcium voltage gates so calcium could come inside. So voltage gated calcium channels open and calcium goes into that. Next thing, the synaptic vesicles fuse with the membrane. So here are synaptic vesicles. The calcium triggers our motor protein to bring the vesicles down and fuse them with this membrane here. Okay. And then once they fuse to the membrane and open, then our neurotransmitter, here's our neurotransmitter, is going to diffuse across the synaptic cleft and bind to our receptor right here. Okay. Then it says current flows across the postsynaptic membrane. So then the ions, once we open up these doorways with our neurotransmitter, the ions that have the ability to flow either into or out of, that's what creates the current, because the current is the flow of a charged particle. Okay, then finally, after we get our current flowing, our ion flow either in or out, then our neurotransmitter that opened up the doorways is going to be pumped, and actually these are our pumps, going to pump that neurotransmitter from the keyhole back into the synaptic bulb where we will reform a phospholipid bilayer membrane around it and hold it in a vesicle until we use it again. So let's watch all those steps, and you need to be familiar with these steps.
Neuron. An action potential occurs in the presynaptic terminal. The voltage-gated calcium channels open and calcium diffuses into the axon terminal. The synaptic vesicles fuse with the presynaptic cell membrane and open. Neurotransmitter diffuses across the synaptic cleft and binds to the postsynaptic receptor. Current flows across the postsynaptic cell membrane. Neurotransmitter dissociates from the receptor and is pumped back into the axon terminal. Click the presynaptic terminal to view the animation again.
Okay, very important. Why did we do all this? And the action of this cell that had this release to it depends on which neurotransmitter was involved. Because remember, we have some things trying to excite this, some things trying to inhibit this, but that's really more dependent on the receptor found on the cell, because the same neurotransmitter could be excitatory or inhibitory. It's according to which receptor it goes to. Now, there are a few neurotransmitters that would just be inhibitory, like GABA, but most of, like epinephrine, norepinephrine, could be either excitatory or inhibitory, and it would depend on which receptor did it go to. Okay, notice here is acetylcholine. By the way, we call receptors for acetylcholine, we call them cholinergic receptors. So acetylcholine, cholinergic. You definitely need to understand that. And you're going to hear that term used a lot if you go on in especially in medical terms. Okay, now notice that the same neurotransmitter, if it goes to this receptor, then it would have an excitatory effect on the cell. If it goes to this receptor, it would have an excitatory effect on the cell. If it goes to this receptor, it would have an inhibitory effect on the cell. Now notice that if it goes to this receptor, it would have a direct excitatory response, because this receptor is one that opens up and serves also as the ion channel, whereas this excitatory receptor and this inhibitory receptor are indirect. In other words, if it bound to that, it wouldn't open up the channel immediately. It would go through a sequence of events before a channel opened or closed. Same thing here. If it bound to this, it would set off a series of reactions, usually using something called G proteins and other enzymes before it would open or close a channel. So again, this would be direct. These two would be indirect. This one would be excitatory. This would be excitatory. This would be inhibitory. Again, you see direct and fast, meaning that it bonds to basically the area that opens up and allows ions to flow through. Indirect or slow means it bonds to this, but it's a series of reactions before an ion channel would open up. Okay, now when we looked at a neuromuscular junction, we were looking at this type of receptor, of cholinergic receptor, which was both direct and fast and excitatory. Okay, so again, that makes sense. When we're wanting to contract a muscle, we want a direct, fast, excitatory reaction going on. We're not trying to inhibit the muscle, we're trying to excite it, and we're also, wanting a fast reaction, not much delay between the time the neurotransmitter is released and the time the muscle starts contracting. Okay, notice nicotine is a mimicker of acetylcholine, so it would cause basically the same type of activity. Okay, well, what if acetylcholine goes to an excitatory, indirect, or slow? What kind of activity would that produce?
Muscarinic acetylcholine receptor, or MACH receptors, are found in the central nervous system and on most effector organs of the parasympathetic branch of the nervous system. Acetylcholine acts indirectly at these receptors, producing a slow, excitatory postsynaptic potential. Acetylcholine is excitatory at these muscarinic receptors, causing neurons to fire action potentials and smooth muscle to contract.
Okay, so in this case, we see that we find these on our central nervous system, our brain and spinal cord. So this type of receptor is receiving acetylcholine, even though it's doing it in indirect and slow. What it's doing, it's eventually allowing positive ions to flow in. Here is our synaptic potential. And notice that our synaptic potential moves away from the initial source towards, here's our axon here. So our axon hillock would be in this area. And if we have enough of this to reach threshold, we will then send our action potential.